So this is just-dna Lite module manager. You can switch here from annotation anytime by clicking module manager in the header. And it can be used to actually create new annotation modules using arbitrary input data, for example, research articles from scientific journals. And AI actually composes the module for you and checks the formats, draws an icon, and basically makes a ready-to-use annotation module. So we will showcase this functionality using some sample articles on fibrosis, and to actually do it, I'll start by attaching some articles on cardiac fibrosis, like these four. And I'll ask AI to compose a module on fibrosis. I want a new module for fibrosis based on these articles attached. Now that's basically enough because it has proper prompts how to compose modules, so I don't need to give it additional guidelines or anything, just I can send like this. So this has two modes. One is single agent, the other is research team. Currently, I will be showcasing research team. I'll launch it because it will take time, and I will tell while it goes. So as you see, well, team run started. And it starts with PI, principal investigator agent, who dispatches the work, so the actual analysis of the articles, to three different LLMs. So these three agents have basically the same prompts, but different underlying LLMs. We use Gemini 3.1 Pro model, OpenAI GPT 5.4, if I recall correctly, and Anthropic, it's Claude 4.6. So yeah, here you can see the calls actually happening. Principal investigator sends the articles to these three underlying research agents with the same task to actually look through the articles and extract the RSIDs or genomic coordinates, which are interchangeable because we resolve this through ensemble, and actually report back to the PI with the findings. Then PI actually compares the outputs from these three agents, and if the output matches, so if it's concordant, then we can expect that the finding from three different agents based on three different LLMs made independently is somewhat reliable. If it is discordant, so one researcher reports some RSID or the other doesn't, and if details somehow does not match for some reason, for whatever reason, it can be a hallucination, a bug, a search problem, then this detail is further investigated, and also we have an additional step that is a reviewer agent. So after the PI agent compiled the output from these three researchers, in the way I described, like by selecting the findings that are confirmed by the majority of the researchers and vetting the findings that have some discrepancies. After that, it submits the draft of the module to the reviewer agent. So this is a critic pattern above this team consensus pattern. To additionally like that results and to filter out potential errors introduced by PI, because there's also an LLM agent is also prone to like stochastic deviations from the process. Once it's done, PI submits the results to deterministic compiler, which actually resolves coordinates and RSIDs to each other. So the coordinates resolved against GRCH38 build, which just-dna Lite uses. And with that, the module is finished. So the single agent research mode, which is an alternative to this, allows you to have like a reduced team of researchers. So instead of three different LLMs receiving this, task, only one agent does it. So this reduced team allows you to save up tokens, and you have like a simplified pipeline with only a researcher and a reviewer. So only a creator and critic. So with team effort, you can also configure how many researchers there would be by adding or removing additional keys. So if you don't have, I don't know, OpenAI key for some reason, you can have like 2 researchers instead of 3, etc. One can also change the underlying models. These are configured in the YAML files of the project declaratively. So if one wishes to actually dig deeper, it can be done. But this can be also used like out of the box by like unprepared user who does not know anything about agents coding or AI patterns, by just asking the machine to create a module based on the articles of interest. So a researcher like who's specialized in medical field, who does not like deal much with computer science, can actually create annotation modules and annotate genomes using only inputs he deals with on daily basis, so research articles or other like texts. So now we can see some progress on the team effort. We see that calls to research is actually completed. Currently, researcher 2, which is ChatGPT, also looks like it completed its task. However, we still wait for the other researchers. As I can see, PI now submitted the inputs to the reviewer. Therefore, we can expect the output shortly. So, on average, this like team run takes from 5 to 15 minutes. It depends on the amount of input that you provide. So with one article, it's closer to the lower limit, and we kept the input to 5 articles at a time in order not to overload LLMs, because in such case they can kind of lose focus and lose details if it's too much input at a time. And well, with 5 articles, it's obviously closer to the upper limit. So judging by this, we can see that PI actually reached completion almost here. He produced the module files already. And shortly we will see the completed module. So yeah, it now validates the produced module against specification. So if it meets the requirements for the application to be able to process it, the modules can technically be created manually because like the specification is also declarative and it can be done by a human researcher, but it's of course a more tedious task. Technically, human researcher can also collaborate with AI here by providing like drafts of the modules and asking to complete the missing parts. It is also totally possible. So currently PI wrote the description for the module. I can see. For some reason I can't expand this one. Because I just got the full reply. So it says it made a module named cardiac fibrosis. It included well, some genes from these articles, 15 genotypes, and well, some RSIDs. It also created documentation, list of studies used for the evidence, and some logo. So now we have this cardiac fibrosis module. We can actually register to the source by clicking this button. So this window on the left, like the editing slot, it holds like the draft of the module you're working on, and you can actually continue working on this. I will probably show the second iteration, but first I want to demonstrate how you can actually work with this module right out of the bat. So this registration button, it sends module for compilation, so it becomes a RK file that will then work via joins. It will become an actual annotation. Okay, so the module actually was registered, and now we have it as an annotation source. So now we can switch to annotation tab, select, for example, Anton Kulaga's open source genome, and go to new analysis. And look, here we have a new module named cardiac fibrosis that we just created. It has some AI generated icon, but it is recognizable. It's like heart with DNA shape, I think. So now we shall start analysis. And we will have a look as it goes. So it now proceeds with 6 modules. We shall look how it goes in Dexter. Now it is complete. So this part might be a bit technical, but you're not like obliged to look at this, and just easier to understand when it finishes. So this Dexter job just finished actually normalizing the VCF. Now it annotates with the modules. Okay. Finished the annotation, and it is composing the report. It finished. I'm closing this. Now we have a success. So let's look at the report. This is a fresh one. Yes. Okay, cardiac fibrosis and remodeling. Let's have a look. Cardiac fibrosis and remodeling. So apparently Anton have two variants for cardiac fibrosis. One is like positively influencing his outcomes. The other one is associated with some atrial fibrillation risk. LLM actually assigned this low negative score. And if we want to read up on this in more detail, we can go to the actual article, which is like the article that I actually submitted for the module creation, so I won't open it. And the RSID, so we can click on this RSID item and have a look like on DB S&P, like what it is about in further detail. So get back to our just-dna-seq interface. And if we actually want to expand this module, continue working on it, we can actually do it in the same module. So the module is currently in the editing slot. And I already added previously 3 new articles while the module was compiling. So I will ask LLM to extend the module with the findings from the field attached. Now simply click send, and the process we already saw it kind of begins anew. Once it's finished, the version of the module and the editing slot will be bumped. So currently it's V1, it will be V2, and we can repeat the annotation with updated module by clicking register again. It will override the module that is registered for annotation. So if it has the same name, it just replaces it. I think we will not show like the whole process all over again because it's lengthy and tedious, but this is enough for understanding all the process of creation of the module, annotation based on module, and expanding the module with new information. One can, of course, always edit it manually if they want. You can download it. The module is a simple archive containing human readable files. So like logo PNG is the picture, module MD is the text that describes the module. The specification file, well, it's a YAML declarative item, and then 2 CSV files, which are studies and variants. These actually describe what to annotate. And last but not the least is the logs with which you can either track what LLM actually did when it analyzed the articles created in modules, or in case some problem arises, these logs can be used to debug the modules if something goes wrong. So that's it for today. Thank you.